Welcome to Matter. How's everyone doing? Excellent. Well, let's try that again. How's everybody doing? Excellent. Thank you. Thanks to the 4017 for bringing some good weather with them, finally, to Chicago. It's like this every day. That's what I keep telling people. So welcome to Matter. My name is Arun Bhatia, Senior Program Director here. How many of you have not been here before? All right, just a few. Just a few. Well, welcome. Just by way of background, this is the commercial portion of the evening. We're obviously a health tech incubator here in Chicago, serving about 200 startup companies and a little over 60 industry partners as well. Majority of them, 2/3 of those are in the digital health IT space, but we have a number of pharma, medtech, health system, payer partners as well. And recently we've been growing our both interest as well as activities in the life science space. We have obviously a number of pharma partners are coming here to Matter, very interested in the digital health and the health IT space and the role that that's playing in the pharmaceutical development, med tech development. But there's obviously a lot of opportunity here in Chicago to develop new therapeutic assets. Great universities, great institutions, great research that's happening here. And so one of the things we're doing at Matter is building up some of our capabilities, some of our initiatives and the resources to support life science entrepreneurs here, meaning drug development companies, diagnostic device companies. So any chance we get, like we have this week at ASCO, to bring in some new voices, some new faces, and hear stories of companies that are making a lot of progress in the life science space, we try and take advantage of that and have them come in and share their story. This is a really interesting time, as many of you know, in the immuno-oncology space. It's obviously a burgeoning space, a number of new therapies. It's becoming actually a very crowded space, but really exciting to see how folks are sort of harnessing the immune system to really tackle oncology. Some of the other converging pieces that we see here at Matter, which are really exciting, is this sort of convergence of computer science, data science that's playing a role not only in drug discovery, drug development, target development, but also sort of operationally in the clinical trial space as well. And then I think from a commercialization side, the environment is really interesting, right? When we think about value-based care and the focus on outcomes, the increased pressure that obviously a lot of pharma folks have in terms of showing real value to patients. We see a lot of our pharma partners reaching back over to startups and to universities to really find innovation to help commercialize. So we are really excited to have featured guests tonight in 47 Inc. It's a clinical stage biotech company out of San Francisco. They're going to share a little bit of their story, share a little bit of the immuno-oncology space, where they fit. We're going to do a presentation for about 20-30 minutes from Dr. Mark Chow, who will share his story as a scientific founder. And then we'll do about 20-30 minutes of a fireside chat, kind of a fun opportunity to learn a little bit more about how they built their company, what they think about sort of the culture of how they build the culture within their company, and how they're positioning themselves with investors and with partners. They've had great success, recently completed an IPO last year, so we'll learn a little bit more about that. Matter.socialqa.com is the great way to go on your phone, your tablet, pose your questions. I am really counting on all of you to go on there. The fireside chat, if we're lucky, is going to be comprised entirely of your questions. So I have a few in my back pocket, but I'm not planning on using many of them. So I'm leaning on you guys to ask some challenging, hard-hitting questions that you'd like to learn a little bit more about. Does that sound good? All right. With that, Mark, can I invite you to the stage, and we'll get your presentation running.
That's good. Thank you, guys. Great. Thanks, everyone. Really appreciate it. So really excited to be here. I think actually, as Arun mentioned, we are from California, so it's nice to see the California weather here in Chicago. We have some of our 47 team members back here. It's really a privilege, I think, to be able to come and talk with you guys in Chicago a little bit about our story. So you're going to hear about the story of how it influences personal stories that we have, patients. We're going to frame this around the immuno-oncology field as well. And as we get the presentation going, I think you're going to see there's going to be a couple of themes that I think we want to get across. I think as Arun mentioned, really in the intro, there's an exciting time really in cancer as the immuno-oncology field has really revolutionized things. So we're going to touch on that as well. So with that, the title of our story is really about how do you innovate in the immuno-oncology field, and how does 47 or CD47 fit into that? Just to remind folks, so 47 is a publicly traded company, so I'll be making some forward-looking statements. These have to be balanced with risks that we have in public documents. Okay, so our story. I'm going to, in the next, again, 20 minutes or so, tell you about the 47 story, but it's really a story about several things. It's about being inquisitive. It's about being bold and humble. It's about having passion to action. It's a story about perseverance and frustration and luck. And it's a story about vision. But importantly, it's a story about this. It's a story about cancer. So before I talk a little bit more, just want to see a raise of hands for folks in the audience. How many people here know of somebody that's been diagnosed with cancer? So family member, friend, coworker, loved one. So I see hands up all around the room. Keep your hand raised if you know of somebody who's passed away from cancer. Okay, so look around. I think it's the same number of hands. I did that just to illustrate the point. I think we all know that cancer is really impacting all of us. It's deeply personal. I think all of us know of patients or families or friends or coworkers that have gone through this. Cancer doesn't care if you're newly married, or you just got your dream job, or you have 3 young kids at home, or you're just wanting to live so that you can see your grandkids born. And I think this is a story that impacts all of us, but at 47, we really embody this. I think all of us within the company have our own stories. We're really formulated around a mission to help patients defeat their cancer. And as we walk through this, I'm hoping to at least impart some of our experiences about why are we doing some of the things we're doing here, how are we developing, and what's our approach for developing new therapies that we think have value. Talk a little bit about CD47 as a science and why we think it's cutting edge in the field of immuno-oncology. And then touch a little bit about what we've seen from going from the lab to an idea at Stanford to the clinic and to impact that we're seeing in patients today. So a lot of people ask, okay, how did 47 begin? And for me, it actually started here. So this is at Stanford University. This is one of the hospital beds in an inpatient unit called F-Ground. F-Ground, again, is a ward in the hospital that treats specifically patients with aggressive blood cancers, mainly acute leukemia patients. The time frame around this was about 2005. I was a medical student at the time, but did all of my training there too as well, clinical training in hematology. Saw a lot of patients that came through this ward, mainly with a type of blood cancer called acute myeloid leukemia, which is, again, is a cancer of the blood. Now, for this treatment, or this cancer, treatment standard is combination chemotherapy. On the best day, in the best case, as a patient, you're here 30 days in the hospital battling infections, low blood counts, fevers, nausea and vomiting from the chemo, hair loss, fatigue. And hopefully 30 days later, you can walk out of the hospital. But in less fortunate cases, the therapy doesn't work, and we really don't have a lot of good options. And you're subjected to more toxic chemotherapy without really a good possibility of cure. And seeing this, and I think for those who are physicians in the audience, it's quite real. And one of the patients that I had the opportunity to take care of really impacted me, I think, at an earlier point, early phase of my career. And this was a patient who had newly diagnosed acute myeloid leukemia, was a woman in her 30s, married, had 2 beautiful girls, probably one of the most upbeat people you would meet, whether in the hospital or not, and had a very aggressive leukemia, had to have multiple lines of therapy, multiple chemotherapy regimens, and always did it with a smile. Whether, everything we threw at her, and you could tell that she was somebody with a mission to really do everything that she could to be there for her kids, for her 2 young girls. And there was a lot of hope at the time because she had an actual bone marrow transplant from her sister. And that's probably the only curative option we have today in this field. Unfortunately, about a year later, she relapsed and her leukemia came back after transplant. And we really didn't have a lot of options there. I think we cycled through a couple of other lines of chemotherapy, and it got to a point where nothing was really working. The leukemia cells were resistant to everything we were giving to her. And I remember a time very vividly where I got the latest report, results, the leukemia is still there, hasn't decreased. And you're at a point as a physician where you have to have that talk of what else can we do? We don't have any other options. And I think as a physician, it's the conversation that you really don't want to have with patients. And I remember actually standing outside of one of these rooms like this, thinking about what I was going to say to her. And went in, came in and gave her the results, said, unfortunately, the bone marrow biopsy doesn't look that good. The leukemia is still there. And she said, well, what do we do next? And at that time, you're thinking, and there's not a lot of options. And it was such a vivid and vulnerable moment in the sense that as physicians, we're used to and have the privilege of providing hope and assurance. And patients give tremendous trust to us to navigate times where it's life and death for them. And at that point, for me, I felt extremely helpless because I knew that there wasn't really any options we could do. And so we had the conversation. I said, there's really not a lot of other therapies we can try. We don't have another clinical trial, don't have another chemo that we can give. All we can really do is do our best to make you comfortable for as long as we can. And she paused and processed and said, well, let's not give up. Let's continue to find things. We try to work with her to find other centers and universities that might have trials open. But unfortunately, she ended up passing away about several weeks later. And to me, that was just a moment, I think, for a lot of us on our team where, gosh, we have to do better. We have to do better with therapies that are less toxic. And the crazy thing, when you think about acute myeloid leukemia, for many patients, we're using the same drugs today that we've used 40 years ago. So you think about that's 4 decades of the same drugs, of combination chemotherapy with its toxicities. So it really dawned on a lot of us, and personally for me, of just asking the question, saying, look, we can do better. We have to do better. And we asked the question, and we said, OK, well, how can we discover therapies that kill cancer cells and spare normal cells? You get rid of the toxicity, but you get the efficacy. Very simple question. And that's how the 47 story, I think, actually started, at least from the perspective of when I came in, was really to develop novel therapies from that perspective. Now, you fast forward, that was mid-2000s. We're now into 2019. The last decade has been really revolutionary, I think, in cancer. A lot of you guys know that probably the most impactful has been the field of immunotherapy, which is essentially using the body's own immune system and mobilizing it to attack the cancer. The best example, most likely, is the advent and discovery of T-cell checkpoint inhibitors. So these are drugs that mobilize T cells. We've seen, actually, this change the way we think about cancer. So there were cancer types where we didn't think about cure before. But now we can start to think about that with these therapies. Importantly, in the culmination last year, so the Nobel Prize in Medicine was awarded to Drs. Allison and Hanjo, who were really pivotal in the discovery of a couple of immunotherapies that were key to today. Now, with all that success, I think in general, there's still a lot of unmet need. So about 25% of patients generally across cancers respond to T-cell checkpoint inhibitors, which means that 75% don't. So we really have a lot of work here to do. I think For folks who know the environment, there's thousands of studies with T-cell checkpoint inhibitor combinations, and we haven't seen a lot of advances since these have really come out. So our view at 47 is one where we think we have to mobilize the entire immune system to really get a full response. And there are two arms of the immune system. So there are one arm, the adaptive immune system, which is what primarily comprised of T cells here on the left. But there's also the innate side of the immune system. And one of the key cells that involve the innate side of the immune system is a cell called a macrophage. So macrophages, they're like scavenger cells. They kind of sense things, and they determine whether something's normal or abnormal. And if something's abnormal, they swallow them up and ingest them. So you can think of this kind of as analogous to first responders, so like firefighters or paramedics, who are first onto the scene and critical in that 5-10 minutes where things really matter. That's in some ways the first response immune system in part that macrophages are involved with. Now, if you take that view and you translate that to cancer, one of the questions that we often ask and get asked is, well, within cancer, there are immune cells that are in there. What is the most dominant cell type that comprises the cancer? And it turns out in a lot of cases, it's actually the macrophage that is the most dominant immune cell infiltrate. This is an example of a picture of a brain tumor where the cells are lit up by fluorescence. Pink cells are T cells in pink, and then macrophages are green cells in red. And you can see that there are quite a bit of macrophages there. Well, we have therapies that activate T cells, but we really don't have therapies that activate macrophages. And so this is where, as we start to think about the science, CD47, which is a target used by macrophages, is actually one of the emerging therapies that we think is a therapy that actually can activate macrophages. And so I'm going to walk you through a video in a little bit to show you a little bit about that mechanism. But I'm going to go back to the same question that we asked in the mid-2000s was, what are targets that you can target on a cancer cell that have minimal impact on normal cells? So at Stanford, this was through our group with Dr. Irv Weissman, Ravi Majeti, Jens Volkmer, Jay Liu, and some others, where we screened a bunch of different proteins and said, okay, what fits this criteria? And it turns out that one of them that was near the top of the list was a protein called CD47. Now, what is CD47? It is a don't eat me signal that's used by cancers and macrophages. So I'm going to, again, now show you a video and walk you through this about how this actually works within the body and how we capitalize on this. So I mentioned that our immune system, we don't just fight infection. We actually sense cells that are normal and abnormal. And so these abnormal cells can, without being checked, can turn into cancer cells and be actually pretty dangerous. And so this is the arm of the innate immune system. This is a macrophage that its job is to, again, look at abnormal cells and be able to sense them, interact with them, determine whether they're healthy or normal or unhealthy. And they do this by doing essentially wellness checks. Again, here you can see here is able to sense that a cell is normal. When the macrophage senses that a cell is abnormal, then its job is to eliminate that and engulf and swallow the cell through a process called phagocytosis. And it does this before really any abnormal or damaged cells can run unchecked. Now in cancer's figured out a way to outsmart this mechanism. And so when macrophages engage with cancer cells, the cancer cell can actually block phagocytosis through CD47 to create a false normal signal that allows them to escape the macrophages and grow. And they do this specifically through expressing this protein called CD47, which basically is able to mask the cancer cell as a normal cell, prevent phagocytosis, so that this cancer cell can go unchecked and the macrophage doesn't recognize it. Now, our ability is we have a therapy that blocks CD47. This is an antibody called 5F9. And the way this works here is that 5F9 actually binds to CD47 and actually blocks this don't eat me signal. And so what this does is you can see the macrophage now can see the cancer cell and see that it's abnormal and actually phagocytose the cell. And it does this, it can do this multiple times, and it can actually re-energize the macrophage to be able to fight cancer. And so this is how 47 really works. And 5F9 is a first-in-class antibody targeting CD47. And I'll talk a little bit more about actually the data that's involved here. And so it's great, I showed you a video, a cartoon, but does this actually work, in patients, in cancer cells, under the microscope? And so this is actually a video that we've taken of human cancer cells in green, macrophages in red, normal conditions, and you can see plenty of interaction with the macrophage and cancer cell, but no ingestion. So these macrophages don't really sense them or see them. But now when we add our 47 antibody that's blocking, you can now see that these macrophages are primed. They're able to ingest the cells. They're able to ingest multiple cells. And they do this quite rapidly. So again, unveiling this ability for macrophages to effectively work. So one of the questions at the outright of this, again, back in the late 2000s was, okay, well, how widespread is this mechanism? We were interested in acute myeloid leukemia for some of the reasons I mentioned to you before. But we had two questions. Number one, first is, okay, well, we can show this in a lab, but is this clinically significant? Does this actually matter in patients? And so what we found was when we looked at patients, we found that patients who had high levels of CD47 actually did a lot worse in terms of prognosis. So this is showing you in blue patients who are CD47 high having worse survival. Now, what was surprising to us was the fact that this mechanism seemed to be prevalent in other cancers. So when we looked at other blood cancers, other solid tumor cancers, we saw the same thing. So we saw that 47 patients who were high did consistently worse. And so we thought that CD47 may actually be a universal mechanism by which cancers evade the macrophage and establish disease. And so with that, we turned to the lab. We actually created multiple models preclinically, showing you two here. One on the left-hand side was a mouse model where we injected human patient leukemia cells. They hone into the bone marrow. And you can see here, this is a control mouse, that all of these blue cells are all chock full of leukemic disease. When we treat with our 47 antibody, 14 days later, the marrow completely empties and the leukemia is eliminated. On the right-hand side is a sarcoma. So this is a solid tumor model whereby you can see this is a mouse lung. And then all of this brown or black dots, this is all metastases, human cancer disease. Again, treatment with antibody, clearance of that. So this was obviously very exciting for us. And we saw this in multiple cancers, but it wasn't just, I think there was a lot of luck involved, but it wasn't just smooth sailing. So we had a lot of struggles. When we went from the lab and then tested, for example, our antibody in monkeys, which is the intermediate step before you go into patients, we found that some of the first few monkeys we treated had a massive anemia, which is a blood loss or clearance of red cells. And this could have been program limiting. We were fortunate and said, okay, well, how do we look at the science? How do we figure out and overcome this challenge? Because we know that there's something here. Fast forward, we were able to redesign our molecule and create a novel dosing regimen that could get around the anemia. And that's been one of the key features that has allowed us to safely treat patients so far to date. So if we move forward now from the lab into the clinic, one of the things we're excited about at 47 is the combination aspects with targeting CD47. So we view a couple of things. I think monotherapy activity with our antibody serves as a foundation to our pillars. In immuno-oncology, we know that monotherapy is really important as you establish combinations. And we have several combination pillars with other agents like anti-cancer antibodies or T-cell checkpoint inhibitors or chemotherapy that we have been able to show actually creates synergistic effects in the lab. And our pipeline is really based on these hypotheses. So we have several trials that are ongoing, spanning blood cancers like leukemia, lymphoma, to solid tumors like ovarian cancer with these different combinations. And we're very fortunate to be able to partner with some of the leading nonprofit organizations as well as large pharma companies to really share in our mission to really investigate the full potential of this drug. And so when we talk about our patient experience, we've treated now well over 290 patients to date. And so far, the drug has been actually generally well tolerated. And we've seen clinical activity in several different cancers, again, in blood cancers as well as solid tumors. These are some examples of 2 publications that we've had in lymphoma as well as in our phase 1 in solid tumors recently in the last year. And I think this being ASCO, we're actually excited to be able to present data for the first time of a combination of therapy for patients with acute myeloid leukemia at ASCO, I think on Monday, with an event to follow for those who are interested. But for us, this is really an exciting time to be able to showcase some of the initial results we've seen. But I really want to just get back into kind of the patient and the patient stories we've seen. So for us, these types of things have been really what's inspiring for us. These are 2 patient scans of a trial that we have in non-Hodgkin's lymphoma in combination with rituximab. This is what's called a PET scan. You can see a patient with follicular lymphoma here. All of this black area, this is all disease in the neck, in the mediastinum area, and then in the gut area. 8 weeks later, that disease is cleared, so that all that's left, all of this is residual normal organ. This is an aggressive diffuse large B-cell lymphoma patient with really bulky disease. Here you can see all these black studs are all lymphoma. Again, eliminated at 8 weeks. What's remaining is just normal organ. And so with that, I again want to go back to this picture where I started, in the sense that when we look at things and we've seen some nice stories, some nice scans, we're always reminded that, again, every day patients are dying from cancer. We've lost many others in that process from even just several years ago. I do want to mention, though, it's incredibly gratifying and motivating that when we do have patient experiences where we've seen benefit from our drug. So I do want to share one story about a patient who's been on study with us, who actually was a patient that came to the University of Chicago. So right here was a patient with lymphoma who had aggressive disease, was actually looking for a therapy called CAR T cells, which a lot of you guys may know. This is a therapy very exciting that's able to energize T cells. But there's no slots available. She was on wait lists across the country. So she came to Chicago, enrolled on our study, was really in bad shape. So had daily fevers, night sweats from her disease, was actually in a wheelchair because her disease was so debilitating. Within a few weeks on therapy, her fevers went away, her night sweats went away. Within a few months, she went from wheelchair to hiking and mountain biking. And so for us, that was a really fantastic story. She was disease-free at that point. The last time we've touched base in with her, she's been disease-free so far. So these are the stories I think that really gives us a sense of hope and inspiration, but also reminds us that we're by no means done. And so with that, just on the last 2 slides, I'll land with this as kind of a framework of our journey. So, 2000s, we made the initial lab discoveries. We've been fortunate to be able to accelerate that in a lot of ways at Stanford, where we've been able to develop a drug that was tested in phase 1 studies, formed the company at 47, was able to initiate some additional studies, fortunate to go public last year to engage in more. But I think the point of this is just to say, while we're excited of where we've been, there's a lot more we need to do. I think the journey is far from over. Our goal really is to get this drug to as many patients as we can. And that takes still a lot more work. And so just in conclusion, I'll say that you've heard that the science is really what's been intriguing. It's definitely an asset for us. But I would argue that probably our biggest asset are the people in the company. So our mission, 47's mission, as I mentioned, is to help patients defeat their cancer. Every single person who's in that company really lives and breathes that. It's actually the reason why, for me, I love coming into work every day. Because regardless of what your role is, you're tied into this. And it starts from the top. I think you'll hear a little bit in our fireside chat. Our CEO, Mark McCamish, really espouses this, sets up deliberate goals to make sure that we put patients first. And that really trickles down. And so I think for us, that's really what motivates us in good times and in bad, to be able to work as a group to accomplish something that we all believe in. So with that, I really want to thank you all for your time, for listening, for hearing part of our story, to know that cancer is relentless. And so we really need to do our parts to really help it. And this is just a small part of, I think, a larger effort that I think a lot of us are working on. So appreciate the time and happy to answer any questions. So thank you so much.
It's a great story. Thank you, Mark, for sharing that. And just by way of introduction, they kept it easy for me. They're both named Mark, so I don't have to remember any names here. So Mark McCamish, welcome. Maybe just to start off, a little bit of an introduction of yourself and how you got introduced to the company.
Sure, Mark McCamish, physician scientist by training, was in academic medicine at University of California, and then transitioned to industry. Started with Abbott Labs, and then went to Amgen, and then did some startups out of Stanford, PDL Biopharma, and then the last, before 47, I was with Novartis, running their biosimilar programs in Europe. And we were the first to launch biosimilars, which is copies of more expensive biologics, allowing for increased access for patients who would not be able to either afford, or their health systems wouldn't be able to afford the biologic medicines. And we had 10 biologic licensing applications filed in the last 3 years that I was present. So very active, productive. I had a lot of experience with the commercial aspects of it, the manufacturing, and regulatory interaction. Since it was a new therapeutic area, the regulatory agencies were learning how to have oversight of the biosimilars. With that, I came to 47, met with Mark. He interviewed me, and that was a great experience. I think, as Mark outlined the science, 47 being named after CD47, and really very exciting science coming out of Stanford. But science is, it's never predictable. So to me, it's the people, as Mark mentioned, because you will always run into barriers, and we've had our barriers. And it's the people that allow you to negotiate through those barriers, and it's based on the values that Mark shared, of the passion to action, bold yet humble and inquisitive, that have allowed us to move that forward. So
That's great.
It's been a great experience.
We're going to talk a little bit more about how the company started and the formation, but I wanted to touch on one thing. I don't know if all of you caught this in the timeline slide. The company was actually formed well after we often think about companies forming in this space. So a lot of the preclinical work was done, a lot of the IND-enabling work was done. 2 phase 1 studies were completed before the company was even formed. Talk to us a little bit about the thinking behind that, how that came about, and if you could touch on one thing, which is we always talk about, investors and partners always come to biotech saying, show us your development plan, show us how you get from point A to point B. How did you think about that in the early days? Maybe share a little bit of that story.
Sure, yeah, so definitely a unique model. So when this was probably 2009, we published the first papers on targeting CD47 in oncology, and there was a lot of interest, I think, certainly in the scientific arena, but also in terms of investors and companies. So we were actually approached about opportunities to license or so forth. And for us, I think, and a lot of the credit really goes to our main founder, Irv Weissman, who many know is a pioneer in the field, really was how can we make sure that we have enough time and effort to understand the science, to de-risk a lot of aspects so that we can be in a position to be able to put this in patients. And one of the concerns was, well, if we license it out too early, is it going to be one of 100 different programs that need to be prioritized, and the first sign of something going wrong, it could get dropped? So we were fortunate at the time to be able to find grant funding. There was an organization called the California Institute of Regenerative Medicine, which was a nonprofit organization that came out of a voter proposition about several $billions to fund stem cell-based research. And we were one of the first groups to get funding. It was a sizable amount of funding. So it was, I think, over the time period, about $30 million that allowed us to develop and humanize the antibody, do the non-human primate studies for IND, initiate the phase 1s. That really got us through that period of the valley of death, where we came through a lot of problems and questions to tackle this. CD47, I didn't mention, it's on cancer cells, but it's also on normal cells. So how do you figure out that balance? And that was key. For us, we were able to then show proof of concept. And at that point, we knew we really needed to accelerate clinical development. That was the opportune time to spin out the company, to bring in private financing to accelerate that, bring in leadership as well. So for us, it was a unique model. A lot of this was done there, but it was critical to our success and ability to make sure we were able to follow the science, but do it in a way that was on a time frame that could benefit patients.
I think it fits into what Matter is trying to do, because you're trying to foster innovation and innovation centers. And one of the challenges is when you're starting something of this nature, when you partner with a pharmaceutical company, they have their own policies and procedures and decision formats and levels of decisions that go forward. And sometimes that can slow down the pace of innovation, of discoveries necessary. And I think at 47, the founders, which are all still associated with the company, their vision is really to have this have an impact on patients' lives. And in doing so, they wanted to maintain as much control as possible for doing so. And the better way of doing that is to advance your program as far as possible prior to partnering with a formal pharmaceutical company, so that you can bring that rapid speed of innovation to the table on an ongoing basis.
I think when you mentioned the 30 million number, there's probably an air of jealousy in the room as we heard that. So
You moved to California.
Well, pass your own tax initiative.
There you go. There you go. Mark, talk a little bit. Dr. Chow mentioned early on in the presentation sort of his vision of the motivations behind the patient centricity, and we hear that term a lot, and we hear pharma using that term a lot of, we need to be more patient-centric. Tell us a little bit about, as CEO, how are you thinking about what that means, but tangibly, how does that actually play into and get incorporated into how you think about running and growing the company?
Sure. When I first was evaluating the company, one of the issues was, what was the values based of the company, and where were they really passionate? And then patients were certainly there and having a purpose around that. And you could see that the goal is really to allow patients to defeat their cancer. And using the term defeat connotes the battle that patients experience. And Mark gave his own experiences. And I'm much older, so I've had many more individuals, family members, et cetera, that have battled cancer. And so we all have that. Interestingly, when I first came in, and 47 is a very scientifically oriented company. There's just a ton of PhDs in the organization. And when I came in, even before I started, I hosted an offsite to really talk about vision and core values and kind of touchy-feely things. And there was a lot of reticence, we have to spend all day talking about these things. But it was really important to explore our own individual views and how we tied in that purpose for patients, and then how do we communicate? How do we move that forward? And we developed our goals and the core purpose that are there, our core values, and we go back to those. So the core values of passion to action, being bold yet humble, and then inquisitive. In the Bay Area, it's very easy to find individuals that have passion, that are bold, and that are inquisitive. It's not so easy to have people that are humble. And so it's part of the core culture that we have, and we hire based on that. our CFO, Ann Rhodes, that's here tonight, really embodies that. She's a very humble individual. Our CMO, Chris Takamoto, embodies that. And so that's a real key component. That allows us to have a purpose around patients that are there. And what we mentioned previously is patient-centric to me is caring about patients, but part of that is listening to patients. And sometimes we as physicians could get caught up in the medical progress that we make. And you can make medical progress which is very hurtful to a patient. So how can you improve their quality of life while improving their medical diagnosis? And part of that is just listening to them as you go forward as well.
And Mark, for you, how did, Mark mentioned when he came in, it was all PhDs. And one of the things that I know challenges a lot of us here is how in a PhD science-focused mindset, how do you almost transition from that to starting a company and growing a company and learning how to tell your story, especially in a space that is very crowded and investors hear a lot of different stories. How did you personally make that transition along with Mark, and what were some of the growing pains in doing that?
Yeah, it's, a very interesting question. I think a lot of us, maybe I'll start by saying that if you asked me if I was going to go into industry, 7 or 8 years ago, I would have said no. I have my academic 40-year plan. I would have been running a lab, probably faculty. And so it was actually an interesting decision about even leaving Stanford and going to 47. And that was really encapsulated, as you can see, by just a passion to want to impact as many patients as possible. But to that point, for me and for others and for us, when we started, we knew the science. We were physician scientists, but we didn't really know much about starting a company, financing, a business plan, let alone development plan. And so we were fortunate that, again, one of our founders, Irv Weissman, he's been very entrepreneurial. He's started a couple of other companies, but it was definitely a transition. I think how do you learn and adapt and understand who you're talking with? When we had our early days in talking to venture capitalists, understanding what were the motivations and points that they are interested in versus an academician versus a patient, you have to clue in on that and understand if there's alignment. So I think it was a lot of learning process. And then just development-wise, which is probably more immediate, even at Stanford, I never developed a drug before, just did labs. So how do we do that? And so we brought in consultants that were really established, that were able to had done this a lot to help us and teach us along the way. I remember when we were doing one of our first development plans for one of the early investment pitches, it's like, okay, well, how long is it going to take to get the drug approved? Well, some of us just sat there and took out a piece of paper and said, oh, maybe like 6 months to a year we'll get this approved, and we weren't even in patients yet. So, we really had no idea. And so I think a lot of it is, surrounding yourself on people who've done it. The composition at 47, what's great is you have, folks that have been green industry like me, science maybe background there, but we've had folks like Mark and our executive committee team that have done this, have been at large companies and small companies and have been part of approvals of many drugs. So that composition allowed us really to learn from each other. And I think that's really the critical piece that allowed us to be successful so far.
That's great.
And I think one good thing is that the story, you're not running a political campaign. So you're not doing research to say what should you say? What do people want to hear? You're really saying, okay, if we're driven for this and our purpose is this and our science is this is our plan and this is what we're going to be doing. And so you tell that story and you attract individuals that then see their own vision in that story, whether it be investors or employees or even physicians working in the field that's there. So part of that is just being truthful and honest to your core purpose and values and rolling it out, and that will attract interest if you're doing something really unique.
Yeah. In addition to sort of the term patient centricity, which again gets thrown out quite a bit, value-based care gets thrown out quite a bit as well, and more so on the provider side, the health system side of how physicians are taking more accountability over cost and quality. But more and more we're hearing about value-based care playing a role in pharma, in drug development, in biotech. again, from your perspective, is that still a term that's just out there? But how do you actually think about that? Is that incorporated in any way tangibly into how you think about building and growing the company?
Well, I think it is incorporated in what we're thinking about in our philosophy and the approach, because again, if you're patient oriented and you really want your purpose to be out there and help patients defeat their cancer, how can you do that? We've been innovative in the science. Mark's participated in that component. I think we've been innovative in the clinical trial approaches that we're using. We've been innovative in working with regulatory authorities. I also think that you should be innovative in the commercialization that's there, and our chief business officer is also here, Craig Gibbs. He and I have talked about various ways. I mentioned my last experience was with biosimilars. So you're bringing in a biologic at a lower cost so that more patients can benefit. Ours is not a biosimilar, it's a novel drug, first in class. How do you bring that in when there is so much pressure on our own economy from a healthcare perspective? Now, drugs are only one small part of that, but it's something that we can look at, and we should be entrepreneurial in that approach as well. So, for example, some of the data that Mark shared with you included a combination treatment with Rituxan and our product. Well, the cost of Rituxan can be high, and then our product could be high. How can you bring that cost down? So one of it is potentially partnering with a biosimilar. So you sell the biosimilar Rituxan at a lower cost, and so therefore your total cost to the patient is lower as well. And it's a way that you can penetrate the market and get your compound out there and really allow patients to benefit from that compound. How can you stimulate uptake of that component? And price is one thing that you can think through. So we've thought a lot about that. It's not something that I focus on with investors because they don't want to hear, well, I'm going to sell it for less money. That's not the point. We want to penetrate the market and ultimately sell more of the product and benefit patients from that. But I think you have to have a mindset where you're innovative on the commercialization as well as all of the other aspects.
So to the point about speaking with investors and convincing investors, I'm going to dive into some of these questions here. What were key points in convincing investors that despite not being first to clinic with anti-CD47, that your approach could be best in class?
Well, we were first in clinic, and then I think we are best in class in terms of the science that was there. But we have met with a lot of investors, and it's something you have to be used to. And Mark's done a ton of presentations. We've all done a ton of presentations that are there. But I think you have to be consistent with your core values, and you will then attract the investors that are aligned with your approach that's there. And the investors we have, they obviously want to make money. That's why they're investing. But the longer-term investors that we have are really, they're caring about what we're doing, and they capture the vision. And when they're investing, it's almost like when I was evaluating, coming into the company. The science is a key thing, but it's the people that will make that science happen. So the investors are interested in the science, but they're also interested in the team and the people. How are you showing that you can operationalize this? How are you showing that you can move this forward? And is it aligned with a core vision that makes sense to them? And we've had a lot of investors that really know the science. And there's probably 4 or 5 investors that we've talked to and really know the science, and they never invested. So it's not just the science. I think science is one thing. It's the people, and it's the purpose and how it's aligned. Yeah. I don't know, Mark, if you want to comment.
Yeah, I think I agree. And I think maybe one of the things, learning through the process, if you're a founder, an early employee, and you're making the pitch on the employee company side, is the earlier you can identify who's across from the table from you and understand what's important for them, how do they view the universe? Like when you think about us, is this somebody that thinks immuno-oncology is important or not? That's going to set the tone. So I think, being able to establish where are their core values that we have as a company, but where do you see that in terms of the stakeholder that you're talking to? The more you can identify and align on what those are, then the more success you'll have to be able to connect. And I think that's been a piece where, again, every investor early on that has been involved, there was that clear connection, and there was that, I get it, I see what you guys are doing, and you guys have a good team. So I think that's important as well.
As you think about the importance, especially in clinical trials, of showing, obviously, outcomes, showing value, One question is, how do you think about identifying which patients will respond better to your drug versus other drugs or combination therapies?
I think this is, Mark outlined some of the science around it, and the wonderful thing is that Stanford had done a lot of preclinical evaluations of the molecule in a variety of various tumors and tumor settings. And then from a pipeline perspective, we have different molecules that we could take forward. And in this CD47 SIRF alpha pathway, you can block this at the SIRF alpha side, which is on macrophages, or you can block this signal at the CD47 side, which is on the tumor. And so you could take both. And we have an anti-SIRF alpha as well as an anti-CD47 that we're both working on. But the science suggested that if you did an anti-CD47, there was a potential to have monotherapy. And one of the questions that you may get to is the immuno-oncology space is quite crowded. There's a lot of activity that's in that area. How do you differentiate yourself from that? So there's a lot of thought going in that if you attack the CD47 because it's on the tumor, and Mark and the founders worked through the antibody approach you would take, and in this case it was an IgG4, which would signal attack by macrophages for the tumor itself. And so there was a chance that you would have monotherapy if you used an anti-CD47 approach. With SIRF alpha, the basic science data suggested that it wouldn't have as much of a monotherapy component. So the studies that were initially done were with monotherapy, which is usual in a regulated environment. The FDA wants to know that it's safe used alone before you combine it. But that signal of monotherapy, which we received, we had ovarian cancer patients and then AML patients who had a response with monotherapy, that was really key to then getting Series B funding moving forward, because that was the way you differentiated yourself in the IO space. So going back to how was that thought through, a lot of basic science that were there, and then the molecule and the design of the molecule. And then Mark brought up something, there's a bit of luck. You set out what you can, and then also you follow the signals that you see, but you have to be lucky as well.
Mm-hmm. Yeah, and you said, excuse me, that the science will do what the science does. I think you mentioned, correct us if I'm wrong, that there was some early sort of anemia in the tox studies that you saw. How did you navigate through that and some of the other hurdles that might have otherwise killed some other programs?
Yeah, sure. So I think it was actually a big important thing that's actually been critical to our success is, so when you think about CD47, okay, it's on every cancer cell, but it's on most normal cells. Your gut reaction is, you guys are idiots. Why are you targeting something that's on normal cells, right? And so in thinking about that and thinking about the discoveries of the lab, but having a development mindset of, okay, how can we get this to patients? It was a lot of intent about the science of understanding, okay, we're seeing the antibody only eliminate cancer cells and not normal cells. Why? Some people will say, okay, good. I don't care about the why, I'm just going to go into that. For us, the why was important. So we had figured out mechanistic ways where there was a second signal that was involved. Instead of don't eat me signals, there's eat me signals that are on cancer cells that are absent on normal. That gives us selectivity. Well, that's important because when we first started in monkey studies, again, as I mentioned, there was a monkey or two that had, again, a pretty profound anemia. We know that CD47 plays a role in red cell biology and actually clearance. That ability allowed us to understand actually that these red cells that were impacted also had eat me signals. So we could develop ways to mitigate the eat me signals on normal cells using a different design of our antibody. And then again, I mentioned this dosing regimen, which is a priming and maintenance dose regimen that allowed us to only clear some of the red cells and avoid clearing the others. And that's been really critical for us. We developed early intellectual property around that. And it was ability about how do you maximize efficacy but minimize toxicity and find that sweet spot. So for us, we did a lot, probably 2 or 3 times more than normal amount of non-clinical studies you need to file an IND. And that was because we knew that these were challenges we're going to have to overcome. We wanted to learn them as early as possible. And again, to Mark's point, we've been fortunate and lucky that those predictions in preclinical models have been quite predictive in the patients in terms of safety. Whereas other programs have had to discover that in the clinic. And so I think being able to understand your compound early in a way that's pragmatic, but you understand why things work. And when things don't work, you have to ask yourself, how can I figure out why? There's been experiences with other programs where had that happened, that company and that antibody would have been shelved. So again, it allows you, do you have enough time to understand the science? You can always figure out the science, but you need the time to be able to do that.
I think it's a great example of a barrier that the company worked through. And other companies had programs that had anti-CD47, ran into anemia, and abandoned the program. And then there were other companies that were trying to design a molecule around the anemia component. And here, I think there was a good understanding that the normal physiology of the red blood cell is a demonstration of the mechanism, because as CD47, anti-CD47 binds on red blood cells, those are cleared by macrophages. That's exactly what you want to do with tumor cells. And the fact that we were doing it with older red blood cells demonstrated that we had the right molecule, it was targeting the right thing, but how do you get through the anemia? So it was a barrier, but by overcoming the barrier, you had a pharmacodynamic marker of what you wanted to do, and by working through that and coming up with this priming dose strategy, really differentiated us from any other company that's there. And so there are companies now, several of which have failed because of the anemia component that they didn't work through, or for other adverse events that they didn't anticipate because of that priming dose strategy. So it really is an area where I think the company, instead of running from that, embrace that and then work through it, and that's kind of the component. And we bring about luck, I think, in my experience, where you can foster luck is that you have people passionate about what they're doing, engaged with what they're doing, and loving what they're doing, because when something comes up that's unanticipated, instead of just saying, okay, I did the experiment wrong, I'll just do something else or go in a different direction, they're really inquisitive and wondering why. And it's that inquisitive nature that allowed us to figure out this and then go through that barrier.
Mark, you mentioned in the presentation there was some intentionality around not seeking out collaboration partners maybe right away. Talk a little bit about maybe what is the thought process or the strategy going forward. There's some questions around how do you envision competing with large pharma companies that have established checkpoint inhibitors like Merck and BMS, but maybe in the broader scale of answering that question, what is sort of the collaboration strategy going forward?
I can start, and then I think Mark can add. I think at the early days, we didn't, even in the early clinical experience, and we're still understanding this, understanding how much value is there with this asset, how widespread can CD47 be targeted. Preclinically, we showed multiple cancers that have been impacted. We know not all of that's going to translate into the clinic. So we wanted to make sure that there is enough control and ownership to be able to understand that. But then to the point about, at a phase where as a small biotech, you only can do so much. You have to understand, where there are capacities where really large pharma excels at and is able to develop your drug in terms of large phase 3 studies, commercialization. So I think these are things we definitely think about, how do we combine the best of both worlds for really getting this to patients. And Mark can comment on, further on that, but I think that's been kind of the mindset.
I think part of this has been in the partnering strategy that we've had thus far. So again, Craig Gibbs did a lot of work at Gilead as basically heading up their commercial strategy. And so we have several clinical development partners that work with us. And in that situation, usually when you do a partnership earlier on, they want to own part of the company or provide equity or others, and you have to make that choice. Here, what we've done is, because we didn't know what the value of the product was, we did these clinical trial collaborations so that they provide their drug, we provide our drug, and then we either split the cost or they'll pay for the cost of doing clinical trials, because it's hard for our company to sponsor all of these clinical trials and the expense associated with it. So part of that is because their intrinsic interest is on their half. If this works, they want to be involved. And so we're able to do these clinical trial agreements earlier on in terms of moving that forward. But in terms of keeping an eye towards commercialization, small company, I think you have to be realistic, but you also have to be pragmatic and opportunistic. The founders want to be able to make this the next Genentech. What's the probability of that? It's pretty low. But at the same time, if you continue to move forward, one way of doing that is to play a role in the initial commercialization. So when you start talking about partnering, you want to play a role in the U.S. commercialization. There's no way that we can do ex-U.S. commercialization. It's a small company, but we could play a role in the initial U.S. commercialization with a partner. So, as we've developed an understanding of the molecule, developed an understanding of the indications we would go in, we can then look for partners and try to craft that relationship so it's a global partner that can take full advantage of the molecule ex-US and allow us to play a role in the US commercialization, so that it builds our capability then with our second molecule, our third molecule, of growing our own commercial organization to build that capability.
A couple of last quick questions, and we're coming up on time. We've talked about telling the story to investors, to potential partners. What has been the story to physicians? How have they taken to the story? We think of physicians as being very data-driven. What has been their reaction to your progress so far?
Yeah, I mean, I think that's one of the fun parts about this and the role for some of us where we interface, with clinicians at various institutions. I think what's helped us is, a lot of us come from the academic background, so we have a sense about what is important, how do we connect, and Arun, as you mentioned, it's about data, it's about science. So, understanding the molecule where there's a clear understanding. Okay, these are some of the folks that pioneered this field. They have keen insight. They're passionate about it. They're taking really rational approaches. So for us, when we interact with clinicians, it's exactly that, and I think that's what's been helpful. And we try to strive for our dynamics really in somewhat of an innovative way, where we select investigators and sites and so forth because we want to partner around them. It's not just, okay, here's our drug, go do our trial. It's, well, what thoughts do you have? Because you're bringing in some element of expertise. That allows us to change our program. So we really like to partner with folks early on to build the program together. And I think that mentality, that collaboration, that understanding actually helps make for a better program where everybody's invested. And so that's been really, I think, one of the things we strive ourselves on is lead with the science, lead with the patients, and create that collaboration. And that's, I think, what has been helpful.
Great. And lastly, as you think about the next maybe 2 to 3 years, where do you ideally hope to be? Well, maybe that's not a fair question. We all know where you ideally hope to be.
The next Google.
What are some of the next big challenges that maybe keep you up at night that you're thinking about?
Well, you will have challenges all the time as data comes in, and I think you have to just keep focusing on your core purpose, help patients defeat their cancer. That means you work through the barriers that you have, and then you do the best you can in terms of doing that. Right now, we're at a great place. We've got a molecule that is functional, that has shown benefit to patients, and we've had increasing interactions with regulatory authorities, so we have potential pathways forward there. We also have other molecules, second and third molecules, that are really pretty exciting to move forward and expand the capabilities. Now you have to balance that. You need the funding to do that. So how do you then acquire funding to be able to move that forward to do the best? And that's where you have to have multiple options because you don't know, what funding is going to be around the corner to do those. And you either scale down or scale up based on the options that you have and the resources you can apply to those options. But to me, it's always keeping focused on the core purpose that's there. And your stock can go down, it can go up. And that's the problem, is that the stock is something that a lot of people put value in. And what you have to do is just ignore that, because what your value is and what you're doing is to help patients. And it's the clinical data, it's the filing, it's the commercialization that will bring that. And over the long run, that'll have a positive impact on the stock. So it's not certain tweets that will have an impact on the economic environment that will have your stock go down. You just keep focused on patients and your ability to bring something really unique to them.
One of the things we pride ourselves on at Matter, when Matter first opened, the entrepreneurs here are very purpose-driven, and I think we sort of hear that in your story as well. So, wish we could have gotten to some more questions, but this has been fantastic, continued success, and we look forward to hearing more from you down the road. Thank you so much for being here.
Our pleasure.
Thank you, guys.
Thank you, guys.
Thank you.